Welcome to Educon. You're listening to Imam. Today, now in this video, we'll talk about eukaryotic cells. And till now, we discuss about prokaryotes and its cell envelope, and also ribosomes and this inclusion bodies. Before that, we discussed about the cell theory, and we had a brief introduction about a cell, the differences between the eukaryotic and prokaryotic cells, and also what are the different shapes of bacteria. Okay. Coming to the topic, eukaryotic cells. The eukaryotic cells include all protists, plants, animals, and fungi. Okay. In prokaryotes, it is represented by bacteria, blue green algae, mycoplasma, and fluoropneumonia-like organs. Same here, it includes the eukaryotic cells includes protists, plants, animals, and fungi. In eukaryotic cells, there is extensive compartmentalization of cytoplasm through the presence of membrane-bound organelle. When you look at the eukaryotic cell, heavy presence of, you can see in the image, by different membrane-bound organelle and non-membrane-bound organelles also present in the cytoplasm. So it is like heavy, that is we call heavy compartment, extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. So eukaryotic cells possess an organized nucleus. Earlier we talked about that eukaryotic cells. In eukaryotic cells, nucleus is surrounded by a nuclear envelope or membrane. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures, and this we'll discuss soon. And their genetic material, as we talked, that it is organized into chromosomes, whereas in prokaryotes the genetic material is exposed. It is not covered by any kind of membrane, so it is exposed. All eukaryotic cells are also not identical. Eukaryotic cells includes plants and animals. Okay, but in the earlier sentence we talked that eukaryotic include all protists, plants, animals, and fungi. But all eukaryotic cells are identical. For example, plant and animal cells are different because plant cells, they have cell wall, plastids, and large central vacuole, which are absent in animal cells. Okay, plant cells, they contain cell walls, plastids, and large central vacuole, which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all plant cells. Okay. Now you can see here we have these are, it comes under the eukaryotic cell. We have plant cell and also animal cells. Okay. Plant cell, we can see there is a nucleus. Okay, it is surrounded by nuclear membrane, and we have a nucleus inside. And we have a Golgi apparatus and plasma membrane, the middle layer cell wall, and outer layer. Okay, the middle layer is the cell wall, the innermost layer is the plasma membrane, the outermost layer is the plasmodesmata, which connect the neighboring cells. Okay, and we have endoplasmic reticulum, which is both smooth and rough endoplasmic, and we have lysosome. We have a low central vacuole, okay, which is absent in animal cells. And we have microtubules, and we have mitochondria and ribosomes, peroxisomes, and cytoplasm. We do have a chloroplast. Okay, all these are present in the plant cells, which is also a eukaryotic cells. In animal cells, you can there is no any large central vacuole. You can see in the image there is no any kind of large central vacuole. So we have microvilli. Okay, you can see here small projection. These are called microvilli. Apart from that, everything is present in both plant cell and animal cell except plastids, vacuole, etc. It's large central vacuole. Now what we'll do, we'll look into each cell organelle from the eukaryotic cells. First it comes cell membrane. The detailed structure of membrane was studied only after the advent of electron microscope in the 1950s. So because before that we are unable to see the see into the details of the cell, what it contains exactly. We only know, we saw only the structural representation of a cell, but after the invention of the after the advent of the electron microscope in 1950s, the detailed structure of membrane was possible. Meanwhile, chemical studies on the cell membrane, especially in human red blood cell, enabled the scientists to deduce the possible structure of plasma membrane. Here, chemical studies where it was studied, especially in human red blood cells. Remember, enabled the scientists to deduce means to extract the possible. This is the structure, approximate structure of the plasma membrane by studying especially on human red blood cells. These studies show that the cell membrane is mainly composed of lipids and proteins. Major lipids are phospholipids that are arranged in bilayer fashion. That I will show you in an image. Okay, how they are arranged. Okay. Also, the lipids are arranged within the membrane and with the polar heads towards outer sides and hydrophobic tails towards inner part. Okay, this ensure this means where the hydrophobic tails are present embedded into the membrane, which ensures that the non-polar tail of the saturated hydrocarbons is protected from aqueous environment. Now we can see in the image here. Okay, see, these are the polar heads, they are the non-polar heads. Okay, then you can see how it is arranged in a bilayer fashion. Okay, here and here. Okay, and we have a integral protein which is completely buried in the membrane, whereas we do have a peripheral protein which are present on either side, okay, of the membrane. And we do have a cholesterol and also sugar which is present in the membrane. They are also forms the components of the membrane. In addition to phospholipids, membrane also contain cholesterol. Yes, I just told you, we do have cholesterol and also sugar. Later, biochemical investigations clearly revealed that the cell membranes also possess protein and carbohydrates. Carbohydrates means sugar. Okay, the ratio of protein and lipid varies considerably in different cell types. Okay, the ratio of protein and lipid varies, okay, from cell to cell types. Okay, in human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipid. Okay, the membrane of erythrocyte means blood cells has approximately 52% protein and 40% lipid. This line, there are some certain chances that it may come in NEET exam, because it's specifically good, because we don't have any percentage and composition of other cell, but specifically it is clearly mentioned that the erythrocyte has approximately 52% protein and 40% lipids. Okay, it may come in a NEET exam, so you should remember this line. Depending upon the ease of extraction, remember this also. Ease of extraction means using how we are extracting the components from the membrane. Ease of extraction, it may be centrifuge, it may be crushing, or different methods. Okay, membrane proteins can be classified as integral and peripheral. Now, for example, see in the image. Okay, so this is when you extract, what happened? These come out easily. Okay, these come out easily, but it will take time and also certain process to remove this integral protein, this area completely. Okay, so here, that is what I said. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. So this will come out easily, this will not come. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. So this is peripheral, which are present on the surface mostly, whereas the the integral protein, they are completely buried in the membrane. So how they are classified? Based on the ease of extraction. To understand the membrane, the structure of membrane, improved model of structure of cell membrane was proposed by Singer and Nicholson. Okay, this is also important line. Okay, widely accepted as fluid mosaic model. Okay, according to this model, what is this model? Who proposed this? Singer and Nicholson. According to this, the quasi fluid means partial, sometimes solid, sometimes liquid. So quasi-fluid nature of lipids enables lateral movements. Okay, remember this, lateral means both sides. Lateral movements of protein within the overall bilayer. This ability to move within the membrane is measured as fluidity. So here, see, we have 2 to 3 points here. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972. One thing, okay, and which is called fluid mosaic model. So fluid mosaic model was proposed by Singer and Nicholson in the year 1972. We should remember this line. Okay, according to this model, the quasi-fluid nature means partial solid, partial liquid. Okay, solid, liquid. The quasi-fluid nature of lipid enables lateral means both directions. Okay, both the direction. The movement of proteins in both the lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. We call this as a fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretions, endocytosis, and cell division. So these are the certain function which is very important because of the fluid nature of the membrane. What are those? Cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. All these functions are okay important. These function means they are from the point of functions. Fluid nature of the membrane is also important for the below function. Okay, one of the most important functions of the plasma membrane is transport of molecules across it. The membrane, as we talked about that membrane, that it is a selectively permeable, means it will allow certain molecules to move across. It will stop certain molecules. It will prevent the molecules not to enter into the cytoplasm. Okay, so membrane is a selectively permeable for some molecules present on either side of it. So means selectively permeable means it will take molecules from inside the cell to the outer side, and from outer sides to the inner sides. Okay, many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Here what happened, the molecules, for example, now we'll go back to the image. The molecule, from here we have certain molecule and we have certain molecules here. So based on gradient, they'll just move across certain, they don't need energy for their movement in or out. For this is inside, you consider this is inside, this is outside of the cell. So molecules move from here to here and here to here without any need of the energy. Such transport is called passive transport. Okay, so now understand, passive transport doesn't need any kind of energy. Okay, neutral solutes may move across the membrane by a process of simple diffusion along the concentration gradient from higher concentration to lower concentration. It means, for example, you have certain now you might have learned about the osmosis. Okay, where from higher concentration the molecule automatically it will move towards a lower concentration. Here neutral solutes may move across the membrane by process simple diffusion. Means the neutral solutes means they don't have any kind of charge on it, because in hydrophobic we have a charge, we need to be so that it will not accept the molecules to pass through the membrane. So hence some kind of neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower concentration. Water may also move across this membrane from higher to lower concentration. Okay, Means water. I mean what apart from other neutral solutes, water also will move across the membrane from higher to lower concentration. And movement of water by diffusion. Movement of water by diffusion is called osmosis. Okay, as the polar molecules cannot pass through the non-polar lipid layer, because the lipid layer, the middle layer means the buried layer. Okay, it is non-polar. A polar molecule cannot pass through the non-polar by lipid layer. They require a carrier protein of the membrane to facilitate their transport across the membrane. Certain polar, for example, a polar means a charged molecule cannot pass through the non-polar layer. Hence, it needs certain protein which facilitate the transport of certain ion across the membrane. A few ions or molecules are transport across the membrane against their concentration gradient, that is from lower to higher concentration. For example, certain molecules, now we talk about from higher concentration to lower concentration. The molecules move automatically. If the neutral solutes, okay, they'll move just like that. Of course, they'll just cross the membrane without any need of energy. Some water also, some molecules move based on the concentration gradient, that is from higher concentration to lower concentration. Same it occurs for the water movement also occur. Okay, movement of water, we talk that movement of water by diffusion is called osmosis. That is also we know. And certain polar molecules, they cannot pass through the non-polar lipid layer. Hence they need some kind of carrier protein of the membrane to facilitate the transfer across the membrane. And also certain few ions or molecules are transported across the membrane against the concentration gradient, that is from lower concentration to higher concentration. So such transport is an energy dependent process in which ATP is utilized and is called active transport. You can take example, sodium and potassium pump. Okay, so what is the role of membrane? It is a semi-permeable. It allows certain molecules to move across on both the sides, and also there is both type of transport occur, passive transport and also active transport. Where in active transport there is energy. It is the energy dependent process in which ATP is utilized, whereas in passive we do not have any kind of energy requirement. Okay, now with this we conclude this cell membrane, and also after listening to the video, please read the NCERT text, and I told you certain words should be remembered completely. Okay, here there are certain words, again I will tell you in this. Okay, here depending upon ease of extraction, okay, membrane proteins are classified as integral and peripheral. Okay, and studies, meanwhile chemical studies on the cell membrane, especially in human red blood cells, enable the scientists to deduce the possible structure of the plasma membrane. Okay, and lipids are phospholipids. Okay, all these are important. Okay, so with this we conclude this. Now we will go into the other section called cell wall. Thank you.